And so with that in mind, now that we are talking about some components, lymphocytes, again, those cells that produce antibodies, they're there to find and recognize foreign things and then allow your body to recognize that something foreign is inside. And so they respond to invading pathogens. They respond to abnormal body cells, such as ones that might be infected by viruses or sometimes cancer cells. If we're not sure what cancer cells are quite yet, we are actually going to talk about that. But cancer cells are going to be cells that cannot stop dividing. We talked about it briefly in integumentary system. Cannot stop dividing. And so sometimes if the cell can't stop dividing, then these lymphocytes recognize it and they're capable of destroying any cells that will just keep regenerating if they're not destroyed at the source. And then there are foreign proteins as well. This is a little bit of a different concept that a lot of us may not have seen. But foreign proteins can be a few things. You've ever heard of the term mad cow disease? That in Britain, if you eat, I think that there's a type of protein that the protein itself, like just amino acid sequences, it's not a living thing at all. It's just amino acids that have randomly linked together, but their presence reacts with cells and causes neurological disorders. And so there are some proteins that you can't break down, and instead they end up becoming something like a virus, something that's not really alive, but can cause problems. Because viruses are just a mix of DNA and plasma membrane components from living creatures, and then they just manage to fuse in the perfect combination to make them very specific to certain tissues. They're like Frankensteins of cells. Many components of different parts came together and then, or can come together and create a brand new thing that now infects and just operates kind of like a machine. And you have to think about it like if the cell was a robot, right? There's all these components to the robot, and then all of a sudden, the robot pieces, so they just get junked out, and then somebody builds a robot out of the junk of many cells, and then that junk machine now can go and destroy other cells. And so it's a little bit of weird paradigm, because even that these viruses, they're not really alive, but they have DNA. They reproduce or replicate themselves in other cells, host cells, but by themselves, they really don't do anything. They don't need food. They don't really produce waste. So they're not really, we don't consider them living because they don't have those characteristics. But they're just the components of cells that have gone crazy and haywire, and new ones generate every so often here and there. And then foreign proteins, the reason this has it, and this is a little vague too, especially like this, because all viruses even have proteins on their surfaces, and so do bacteria. But that's essentially the premise behind how lymphocytes recognize it. If you have a little bug inside of your blood system here, and they're going to have little proteins on their surface, and so lymphocytes recognize them and say, hey, this is foreign. Let's destroy it. And now lastly, a new word for us, lymph. Lymph is essentially fluid that has entered vessels, and let's get into that now. This image is a little it's not really covered as extensively in a lot of anatomy classes across many campuses, I noticed. And so this is my attempt I was able to get in the summer to manage to get it in with the material. And so these vessels here are running alongside your blood vessels. You maybe never really see this, but you have a whole entire vessel system called the lymph system. And these lymphatic vessels are going to have these tiny little grooves called nodes. And they're going to be these little bumps, and we'll talk about them in a second. And they're going to be organs that we might have heard, and some of you have asked me throughout the semester sometimes. Tonsils is an example. The thymus gland is going to be this one, not to be confused with the thyroid. It looks very similar. And then the spleen that sits on the left-hand side, very posterior in the abdominal cavity. And then you're going to see even your digestive tract as food is traveling through later in the semester, you're going to see as it's being absorbed that you have these lymphatic vessels or this lymphatic tissue that's associated with your mucosa. And that just means the inner lining. This sounds more confusing than it is. Epithelial tissue. I'll teach you all this later. But really, all it really is trying to say is that there are other tissues. But for right now, do not worry about this right now. And then the appendix is going to be also considered lymph tissue. And then finally, your red bone marrow is technically considered lymph tissue. Why would we consider red bone marrow lymphatic tissue? What stem cells do lymphocytes come from? Anybody remember that? Right? And so lymphocytes come from lymphoid stem cells, which came from hemocytoblasts, which are found in red bone marrow. And so since these generate from or differentiate from these, that's why we consider red bone marrow even a lymphatic tissue. And more on all of this as we move on. And so what do I want to add? Because I'm going to try to cut some stuff off because of time. And so let me read it through very quickly. And so let's add this, and I'll take off the vessel names after. That when we talk about something being primary, that's going to be the site where the lymphocytes are formed. And that includes the red bone marrow. We know that one already. Thymus gland, we don't know. We don't care. And then finally, secondary lymphoid tissues is where they get activated. Activated. Kind of like an on and off switch. And all of this will come up in a second with better images in a bit, so don't worry about it right now. And we'll keep it light. And let's do that. And let's move on. And so all of these vessels, again, are just called lymphatic vessels, and that's all this last note is trying to say. And typically, you'll hear me say either lymphatic vessels or lymphatics to imply the same thing. And so that's really that last note there. Now, this should look very familiar now. This is going to be the capillaries. This is where things are traveling through, right? All those blood cells are making their way across, dropping off the oxygen. Within the plasma, within these blood vessels, all those yummy nutrients are exiting the capillaries and serving and feeding all the tissues that are there. As a reminder that there is a fluid within this space that even though it's two-dimensional, you have to imagine a fish tank when you look at this, that this is completely filled with fluid. We call that fluid interstitial fluid, but we know that already, right? Interstitial fluid. And as you can see, even though we don't talk about it when we're in blood vessels and capillaries, you have another bit of a capillary network that your lymphatic vessels create, and they place these vessels inside, and the volume that's in here makes its way into these vessels. And these vessels, once the fluid is inside of them, it's called lymph fluid. And so lymph fluid essentially becomes interstitial fluid that has entered the lymph vessels, and that's what lymph is. Lymph is, fluid within lymph vessels. That's what lymph is. And that's the difference between the word interstitial fluid and lymph. Is that interstitial fluid is a shared fluid between all the populations of cells there, between the capillaries of the vessels, of the veins and the arteries, the venule and the arterial. And then when we've entered inside of the lymph vessels, we become lymph instead of interstitial fluid. All right. We call the little tiny capillaries lymphatic capillaries if they're associated with the capillaries. And the big tidbit about them as well is that this fluid, it's going to end up only in one place, and it's going to end up being dumped or drained into the veins. And so we have this note here. The lymphatic vessels carry lymph from the tissues to the venous system. This word peripheral, again, means the outer tissues. So carries lymph from the tissues to the veins, That's what venous system means. These capillaries in general, if you were to compare them, they are closed at one end. By comparison, the capillaries are always continuous, right? They always went onto the other part, their partners, right? The veins, the arteries, and the veins are linked together by capillaries, but you'll notice that these capillaries are dead ends. And so flow typically tends to be unidirectional, or meaning that it goes in one direction. And they tend to be larger. They have thinner walls. And they are incomplete. If you look at the inside, if we zoom in on these vessels, you'll see that their lining, this inner lining, is broken if you were to compare it to capillaries. And so this is what allows mass transit of the interstitial fluid to go into the lymph vessel and be called lymph fluid. And so lymph flow refers to the fluid going in whatever direction it needs to go. And you can see that even the lymphocytes are there in case there's a little dangerous little pathogen that makes its way out here. Then when it goes into this fluid, the lymphocytes are waiting for it so that it doesn't progress. You may have heard that cancer, if it makes its way into the lymph nodes, we all hear that. Maybe we've all had some family members or some experience that somewhere. And the reason it's dangerous for cancer cells to enter the lymph, because let's say that your lymphocytes don't recognize it as dangerous, and now you have a cell that's going to be able to drain throughout your entire body. If it made its way in here somehow, like let's say it's breast cancer, and then one of those cells makes its way into the lymph flow, and then it travels, and it can't control its division. It can end up somewhere else, and then it can start developing cancer out of a different part of your body. And that's what metastatic or metastasis, that's what it is. It's when the cell travels from a site that it was causing cancer, and it makes its way somewhere else. The lymph system is a mechanism by which cancer cells can actually metastasize throughout the body. And so that means that they can spread through the lymphatic system. And so that's why getting infections in your lymph system can be a little bit dangerous, because that means your lymphocytes drop the ball, and then all that danger can passage through your lymph system pretty easily if your lymphocytes aren't doing anything. And the good news is that these cells are the same thing that we've been discussing, is that the inside of the vessels of blood vessels were called endothelia. You're even seeing one little simple squamous cell right here. And then, same concept, this flat cell, one cell and another one adjacent, made out of simple squamous cells, so they're simply called the same repeated word, endothelial cells. And they overlap, and this is why they can even act as a valve. And do I have a better picture of this? I thought I did. I did not. Only in my head, I guess. And so when you look at this segment, you can see that there are some areas here that have a valve, that you can see that these extensions of the cells come together within that flow. And what do all valves do? They prevent backflow. They don't let things go in that direction. That's why we say that they are unidirectional flow. You can even see all the arrows are draining into just one space. And the valves help with that. They prevent backflow. And so overlapping cells can create these valves, and valves only allow one-way entry. And these valves are large enough to allow certain things to go through, solids to go through, and some of those include proteins. But this, in turn, can allow the entryway of viruses, bacteria, and cell debris. And so your white blood cells are exposed to these things. When you ever wonder, like, oh, how do my white blood cells defend me? Again, anything that enters your body, usually it's all going to enter through your digestive tract. If you eat something bad, then that means that maybe it made its way into the blood vessels. But then those bacteria get out into these spaces, whatever tissue they're trying to infect. And those cells, those bacterial cells may either infect the cell that's there, or if we're lucky, they make their way, they're just going through the interstitial flow, and then make their way into the lymph flow, and then the lymphocytes are waiting for them and destroy them before they can spread throughout the body. And so that's essentially how your immune system works, is that you have these little defender cells patrolling your capillary and the fluid around them for all those little dangers. And so that's why I have this note here. That's when they're exposed to our white blood cells, and more importantly, lymphocytes, right? That's the theme of today. But just keep in mind that there are other white blood cells that get exposed to dangers because of this very movement of solutes and bacteria. And so the last thing to add, this one-way flow in the valves just prevent things from going back. It's really meant to just go, if something dangerous enters, it's meant to experience a gauntlet of white blood cells so that it can not survive and infect your body. And so with this one, I'm going to have to briefly move on to something else. And this picture here. You might want to switch here. And so before I talk about those previous images, I think it's a little bit easier to understand the drainage system. That this image here is indicating what exactly or how this fluid from all those capillaries, where do they get drained? And you'll notice that there's an entire side on the left-hand side that is completely drained. That most of the body is completely drained only on this left-hand side. And then on the right-hand side, everything on the right is only taking care, this drainage site takes care of everything, which is about, I would say, what's that, 1/4 of the body? A little bit less, right? 1/5 of the body, like 20% of the body goes into this side. And if you look at what we're talking about, this is a duct system, and this is the right lymphatic system. It's not really given a unique name, just right lymphatic duct. And so if we look at what this image actually is trying to explain, is that all the fluid is going to drain on this side. Most of it on this left-hand side is being drained through the thoracic duct. And so this thoracic duct is going to be at the beginning of your thoracic cage. You can see rib 1, rib 2, rib 3, rib 4, and so forth. How does that expression go? And so on and so forth, so on and so forth. And then this thoracic duct is going to drain that lymph fluid right back into the veins. And then the veins are going to circulate it again, because what major vessel is this? Our superior vena cava. And it's going to go into the heart. And so you can see plasma and lymph fluid are almost identical. When you look at the inside of here, you're thinking about what is actually being exchanged in here. It's all the contents of plasma that was released in the capillaries. And so you can see those substances, the ions, some of those proteins, they're all going to enter back into the veins, and then the heart circulates all of those things all over again. And so the take home from this image is that things are progressing upwards into the thoracic duct, and also from the right duct, the right lymphatic duct. And again, it's disproportionate as to how much liquid each side is draining. That's one of the questions I'm going to ask, right? Which one's receiving 80% of the body's lymph fluid, and which one's receiving 20%? I can either ask it either way, but it's an important fact to know. And the name of the one on the left is thoracic duct. And we're going to move on from here, and depending on how much time we have, where I'm going to end is I want you to know the names of these. If not, I'm going to add it to the lab exam when we get there, because they're already in your lab list. And so we'll add it to the lab list, and we'll learn it then. But for now, we just need to know drainage of the thoracic duct and the right lymphatic duct. And know that they're disproportionate and which side drains more. And if there's time, I'm only seeing about 30 minutes left in the class, so then we'll add this if there's time left. But if there isn't, we will not. Anything I'd like to add here? And lastly, I bring these up as well because in the lab exam that's going to come up, not this muscle one, but the next one, these nodes are going to be there as well. And as you look through the models throughout the weeks, because you're going to be learning these muscles, for example, this is our sartorius, this is our rectus femoris, this is our vastus lateralis. And so these muscles are going to be muscles we're going to see. And you're going to see as you're exploring those muscles, these little segments that have a lot of these little nodes, these little bunches here. These are called nodes, lymph nodes. And this is what swells up when you have infections. Sometimes when you see like, oh, the doctors will check if your nodes are swollen, and that's what they're checking. These nodes, you have them both in your cervical region, and then you also have them in your inguinal region, right? That's why they're called inguinal lymph nodes, because they're running along that inguinal region towards the groin. And then that's really it. You have some deep ones and some superficial ones. And so I bring these up because I want you to know what they are. And just keep in mind, these little dots are nodes, and the rest of it, vessels. There is nothing else I need you to know here. Don't worry about this text. I really am just trying to prepare you for the lab exam that's coming up after this next one. And so knowing this will give you a head start. And so we go back to where we were to finish off anything we needed. This last image here is a pretty cool image, a microscope image of the valve. The squamous cells, the end go here, ends go out here. And so you're seeing the valve, and you can see that if fluid is flowing, it would never backflow because the valve will not allow it to. So things go and flow with the pressure, and then they never return back through that valve. And yeah, so these last two things are the only important things I need you to grab right now. Those last ones, if we make it to it, we'll get to it. But if not, this is the main importance, valve prevent backflow. And then now this last fact we mentioned in the earlier respiratory packet, in the very beginning, it starts off with that. And now you might understand why I kind of have this weird split between these. But otherwise, the vessels, the veins themselves have valves as a reminder. Arteries do not. And the thing that helps the veins or assists the veins in circulating blood is using muscles. Right here, you're using your calf muscle, your gastrocnemius. And when you use it, you encourage blood to move towards the heart. And so it's the same concept with lymphatics. If they have valves, that means that they need those valves to prevent backflow because there's not enough power for them to just keep flowing consistently. It's kind of like their flow goes like this. They don't have enough power. Like the arteries, they send the power all the way down to your toes if they want to in one shot. But veins, this is the movement of blood in veins. This is the movement of blood in vessels. It's moving stepwise. And so muscles are required or assist with the actual movement of fluid. And you can see veins and vessels run along each other. Even though we didn't really see it throughout the whole semester, we always saw green and I kept saying, ignore it right now, ignore it right now. But you can see how closely embedded it is with blood vessels because again, they all share the capillary network. Their job is to regulate this capillary network of volume, of nutrients, of dangerous pathogens. And that's from now on, I know you're probably used to connecting red and blue. Now you should always expect to see red, blue, and green for the rest of your careers. And so we move on because I don't think we'll have time to finish this, and I'd rather you get to bigger take-homes. And so we move on to our lymphocytes. And let me see where we're at very quick. Cool, we'll be able to finish the rest. And so the lymphocytes now, remember that they are not the most abundant neutrophils. Remember our acronym, never let monkeys eat bananas? Lymphocytes make up second place of all the white blood cells as far as abundancy goes. Most lymphocytes are located in lymphatic tissues. This is where we expect to find them in the body. And there are three classes of lymphocytes. There are T cells, B cells, and natural killer cells. You're going to see the word cell-mediated immunity. This essentially means it's going to control the intensity of the immune response. Cell-mediated immunity controls the intensity of the immune response. Know their definition's as simple as this. B cells they produce antibodies. And remember that antibodies always seek what? Antigens. And we saw this when we talked about, and I'll go ahead and show pictures to serve as a little review for this segment, that when we talked about blood typing, that every individual, if you're type A, as a reminder, you have antibody B, so there's no interaction, you're safe. But if this person who has surface antigens, and again, antigens are just surface proteins, and these proteins, again, if they find an antibody, then they react, and then they cause blood clotting. But in the case now of what we're talking about, let's switch the image now, and let's create little bacteria now. We're going to change, these are little bacteria now, little evil bacteria. Little evil bacteria, and they have their little surface proteins, and that's what they are now. And so your B cells produce antibodies that are sensitive to those bacteria. If you've ever been infected once, the B cells are responsible for creating these antibodies that will always seek little pathogens. That's their job, is to create antibodies for dangerous things. And even if you think about it in the context of vaccines, now it's pretty relevant to talk about B cells in the context of vaccines. When you have vaccines, you are introduced, like what is a vaccine? I'll give you one of traditional because it helps you understand a little bit easier what's going on. That let's say this is our dangerous bug. This can be a virus or a bacteria. And what we do with vaccines is that we break it apart, and let's say we break it into little chunks of virus or bacteria. And what we do is that we isolate something that's usually pretty important for them to infect you, like maybe one of these surface receptors. And then we separate it out, and then we just do it for 1 million bacteria, and then we're going to introduce a shot or an injection of nothing but that little dangerous piece of the virus. We're just going to grab a bunch of it, and then we're going to put this in your body. And it may seem like, what? Why would you do that? And it's because this is useless without it being in the proper configuration. This is like a human is as, I'm dangerous, so a person can be dangerous, but if I chop off my hand and I throw my hand over there, my hand isn't going to be able to do anything to you because it's not attached to the brain, right? It's not attached to the organism. It's just a hand by itself. Same concept is that instead it's a plasma membrane protein, for example. One of those protein channel proteins, or sometimes they have unique glycocalyxes, or those proteins with sugar on them, and then they recognize the cell they want to infect, and then they go in. So instead, we isolate that without the dangerous stuff inside, and then your body then says, what the heck is this? And then the B cells will then produce antibodies. They will produce little drones exactly the way that we saw. It's a little bit complicated as to why they look that way, like the one I showed you on this picture. They don't all look like this. Sometimes they just look like this, just like this part. And so your antibodies can either look like this or they can look like this, like little pieces. But that's essentially what B cells do, is that they produce antibodies that will always look for, now let me use a different color. Now that we introduce the dangerous thing, this into your body, your B cells produce antibodies that are very, very, very specific to it. I'm going to try to draw it up here. Very, very specific to it. They produce these antibodies, and now they're circulating in your blood flow. So now if the real thing enters your body, you have antibodies there that are already there to detect it, and then they can trigger the destruction of that dangerous thing before it can infect you. And so that's the premise behind vaccines, is to introduce you to something dangerous so that if the real thing comes into your body, you already have defenses for it. And the major cells that play a role in that memory are your B cells. And so this antibody-mediated immunity is sometimes also going to be what we refer to as our memory immunity. Something that will be asked in a micro class or immunology class. Memory. Yeah, I'll just leave it at memory. And that's what gives you memory. And so most cases, you might get infected. You don't have vaccines. You get infected by something. Your body will remember the infection and create the antibodies for it anyways. But, some bacteria or some species will kill you before you can develop the antibodies to defend yourself from them. And so the next thing to add here is that B cells, That B cells and natural killer cells, these develop in the red bone marrow, and that's it. And so B cells now, what is a plasma cell? If you think about dangerous bug A, and I draw another bug, dangerous bug B, and I draw another bug, dangerous bug C, and these are just 3 different organisms. Could be 3 different organisms. Maybe it's the same one, but something slightly changed on the surface, making it way more complicated. But just think about bug A, bug B, bug C. They're different little bugs, different pathogens, different viruses. What a plasma cell is that once a B cell has said, hey, A's in here, my job is to produce antibody A for little dangerous bug A. And so it kills A. And so what it does is that it'll evolve or differentiate into a plasma cell, and then a plasma cell's job is to only produce this antibody. And so if you got infected by bees at some point in your life, then a B cell will turn into a B plasma cell and only produce antibodies for B. And then the same thing goes, you got infected by another thing. It's completely unrelated, like C, and then your B cell will turn into a plasma cell for just C. So what a plasma cell essentially becomes, it's a mega producer of antibodies, and that's it. It's a mega producer of antibodies. Your B cells get programmed to create the antibody, and then they transform or differentiate into plasma cells, which will pretty much be producing the antibody for that very thing that infected you. These plasma cells are the reasons that some vaccines, you only need to get them once every 10 years, one every 7 years, just varies, and then once every year. The difference is because these plasma cells wear off eventually, but some of them last longer than others. And so that's kind of a little bit of background behind your vaccines and antibodies. And so with that, lastly, we have our natural killer cells. These are doing exactly what a patrol dog does. You know, they destroy or neutralize whatever's dangerous. And so they're the patrolling security. And they destroy everything that's marked for destruction or anything foreign. And that's going to be our natural killer cells. And now we move on to our T cells. Our T cells are called T cells because they develop in the thymus fully. This is where they really finish their developmental process, or the thymus essentially controls or regulates their differentiation, because you can see that there are 4 kinds. But remember, lymphocytes come from the same source. All of them come from lymphoid stem cells. All of them. But they differentiate. If they stay in the red bone marrow, they're destined to become B cells and natural killer cells. If they migrate to the thymus, they are more than likely going to turn into T cells. And a lot of these are going to be pretty easy to understand. For example, cytotoxic T cells, these kill foreign cells. They're toxic to other cells. Then you have helper T cells. They activate other lymphocytes, including B cells. They're the ones that actually recognize a bug, and then they go and find B cells, and then they program them. It literally is like what you would imagine. It's like they're like the ones that scour, like they see something dangerous, and then they find a B cell, and then they say, hey, I found this. What is it? And then the B cells read it, incorporate it, and then they make an antibody and produce it for the body. But T cells, they're the ones that they find something dangerous, they'll grab it and say, what the hell is this? And then the B cells, the B cells can detect they're bound to something, and the B cells read it. So that's what I mean by activate it. And you might encounter this in immunology, but that's a simple gist of it, is that the helper T cells, they activate the other lymphocytes. Regulatory T cells, they're going to regulate the intensity of the immune response, a little bit more beyond the scope of this class, but just keep it simple. Regulatory T cells regulate the immune response. And then finally, memory T cells are going to respond fast to invaders that have infected them before. And why did I put that DD there? Sorry, I'm not remembering why I wrote that. Not sure. Respond to DD invaders. What did I mean by that? I'm just going to put previous invaders. I don't know what I meant by that. And so once they've detected something, they create more T cells that are very specific to that dangerous thing, and they actively kill it. Because you can see that these B cells, they produce antibodies. I never once said that these B cells kill things. All I said is that they create antibodies to find things that have infected you. And things that get marked with antibodies will get destroyed by memory T cells, cytotoxic T cells, and even natural killer cells and macrophages, which are not on this page. But, there's a large scope of complication. But the take home here is if I tell you that something responds to in previous invaders or destroys things marked with antibodies, more than likely, they're just actively destroying them. And so what do we need to know about these cells? Cytotoxic T cells attack foreign cells. This can be bacteria. And are cells infected by viruses. And their attack, again, they're toxic to bacteria, to cells, other cells, whether it's bacteria or not, or your own cells if they're cancerous. And they require direct contact. Big difference. And they're the ones that regulate the intensity or the severity of the immune response as well. Helper T cells, they do exactly what that sounds like. They stimulate the activation of T cells and B cells. And this is that point that I meant, that T cells introduce the dangerous thing to the B cells, and they activate our B cells so that they can produce antibodies. So the B cells don't know what to do unless the helper T cells to show them what's dangerous. And that's why they're called helpers. Regulatory T cells, they just simply regulate the immune response. There's a huge study on this, but honestly, it's just easier to learn this. There's enough on this exam. And then memory T cells, they respond to antigens that you've already encountered. So they remember things that have infected you, and then they actively seek and destroy them if they've entered your body so that you don't get infected. And so these B cells, just to touch on the same thing, if B cells get stimulated, and again, B cells produce antibodies, they will eventually become plasma cells that produce and secrete antibodies. And here I just put an insane amount, just to imply they're the main producers once they've turned into plasma cells for whatever specific danger has entered your body. And they are responsible for an antibody-mediated. We understand that if they produce antibodies, anything that marked by antibodies triggers other cells to come and destroy them. And so these antibodies are circulating in your plasma, always attacking foreign things. And so you can have antibodies for cancerous cells. You can have them for bacteria, for viruses, and let's write that. Weak antibodies for cancer cells, bacteria, and viruses. And there are some diseases where you produce antibodies for your own body parts by accident. Usually it's a genetic mutation, kind of like the way that cancer forms, like your DNA gets messed up so the cell can't function properly. So if these cells' DNA get their DNA gets altered in any way, they can accidentally create antibodies that kill your own body. You ever heard of autoimmune disorders? Those are referring to the fact that your white blood cells or your immune system is attacking your own population of tissues. And then antibodies, as a reminder, I mentioned macrophages a little bit earlier on in the earlier packets, where we talked about monocytes and that they turn into macrophages, so they enter tissues and become macrophages, and they engulf things that are embedded or marked with antibodies. And then you can see that we also covered that thing here. We talked about lymphocytes. Now I told you we're going to make full circle here. That lymphocytes come in different, right? They come in, that's lymphatics. So sorry, here. They produce antibodies, the B cells, and those were also a form of globulins, and they seek foreign substances. And then we call foreign substances In sciences, we call it antigens. Or really, antigens are things that bind to antibody. And that's actually the simplest definition I want you to hold on to. Things that bind to antibodies. You can genetically engineer antibodies to seek absolutely anything as well. So you're going to see a lot of genetically modified antibodies as treatment. So that even some of vaccines are already antibodies. Even the COVID vaccine that we as the general population didn't get, you hear when the president got sick the day of, he gets the antibodies. These are a little bit more expensive to get. And so typically, you know, depending on your insurance or who you are, you got the antibody treatment because it's going to kill COVID immediately because it's already there circulating and it triggers your immune response. I don't know if you all heard that. And the natural killer cells, they do exactly what they sound like. They kill foreign cells, virus-infected cells, cancer cells, anything that can harm you, they kill. And they're always just patrolling. That's really it. They don't have any antibodies to secrete. They just patrol around and then destroy anything. They recognize what's foreign, typically in a few ways. And this is a nice little summary. Hemocytoblasts, right? You have your two stem cell populations, myeloid, lymphoid. We haven't talked about red blood cells and the other white blood cells that are made in this whole topic. Really, the key has just been the lymphocytes. But the lymphocytes go into two directions. They can either stay in the bone marrow to differentiate into B cells or natural killer cells, or they stay within the thymus or go to the thymus, and then the thymus is responsible for turning them into whatever they need to be turned into. And so that's what this point is. If it stays in the bone marrow, those lymphoid stem cells, they will become B cells and natural killer cells. And again, lymphoid stem cells become the various T cells, more T cells, and then they do the different types of immunity. Cell-mediated, antibody-mediated, and immune surveillance. Make sure you know which cells are responsible for which. And then to finish up, let's see, I got 8 minutes. What is more important? I'm going to add 2 things, I think. These last 3. This one, I'll bring it back when we get to digestive. So let's skip nodules and organs, but I do need this little first part first. And so now when we talk about lymphoid tissue, we see groups of things. I keep saying, oh, look at these nodes, but I haven't really talked about what's in them. And so these nodes, I'm not seeing any here. Yeah, there's some here, nodes, these little bundles of tissue. There are bundles of lymphoid tissue, and the simple definition of a lymphoid tissue is that it's just a sac of connective tissue that pretty much has lymphocytes. That's it. They're just little segments that have a bunch of lymphocytes. So as you look at a lot of these photos, let's use this one for example. This is a node, one of those bumps that I keep showing you. These little bumps. And then you can even see them on any of the images, right? We've been seeing them all around. There's more on this side, more of the same, actually, it's the same image. And these are the nodes that I'm talking about. There's these little bumps. They're aggregation of lymphocyte. In other words, they're just held together by connective tissue, and then they have a bunch of lymphocytes, and that's all those structures are made of, and you have them throughout the entire body, wherever you have those little nodes. But then also, this is also a little aggregation of lymphocytes. This is the thymus. This is where those T cells develop, or this is the organ responsible for the development of those T cells. And then you have the spleen. And the spleen is going to be the last one, so we're going to talk about these 2. And then these little nodes. These are little bundles or groups of lymphocytes. And so that's all a lymphoid tissue is, or a nodule. It's just densely packed lymphocytes in an area wrapped around by connective tissue, areolar tissue. And remember, this is a connective tissue. And then those are referred to as nodules when it's just the connective tissue, but there are also larger ones, such as the spleen and thymus. These are considered organs, lymphoid organs. Big difference between nodules and organs. And what's the difference between a nodule? Has no capsule. Oh, it's already there for you, sorry. Here, has no capsule. Wrong thing. Has no capsule. And this one has a capsule. That's the big difference. But otherwise, definitions for them are exactly the same. Connective tissue wrapping around lymphocytes. One has a capsule, one does not. That's the only difference between a nodule and an organ. And so these, since we ran out of time, I'll put these on the next one, on the next PowerPoint for digestive. And so nodes are going to be these structures here that, again, contain an aggregate. And I don't want to rush. I hate to rush and just describe it, so I'm going to stop.